We didn't talk about dendritic cells yet. We just briefly mentioned on the dendritic cells. And those are very, very important cells of your innate immune system. Okay? And you have several subcategories of dendritic cells. Those are part, again, of your innate immune system cells, right? So they are deriving from your myeloid lineage, as with most of your other innate immune cells, right? And they develop in the bone marrow. So these are coming from your red bone marrow. All right, cool. All right. And that is from hematopoiesis, right? From your hematopoietic stem cells, also called pluripotent stem cells. Okay. All right, so different types of dendritic cells. You have your classical dendritic cells. Okay, their picture here. They kind of like all look the same, but they have different jobs. Okay. So your classical dendritic cells, they tend to hang around on in the epithelia. Okay, epithelia is like type of skin tissue. Okay, so they catch pathogens that go through the epithelia, right? And once they catch these pathogens, as we're going to see later how these cells work, they are really fascinating. Okay. They catch the pathogens, and then they travel from the site of catching the pathogen all the way to a lymph node or a secondary lymphoid organ to present it to T cells. Okay, so your dendritic cells are really the most effective cells that are linking your innate immune system to your adaptive immune system. And dendritic cells are really, really important in this job because they are really the only ones that can effectively activate naive T cells, meaning T cells that never seen an antigen before. Okay? There are other cells that can do that, but not as effectively as your dendritic cells. So let's keep that in the back of your mind. Okay? So there's a lot of story with the dendritic cells that you're going to come back and look into it. Okay, and then you have another type that is called plasmacytoid dendritic cells. Now, look at the name of this thing, plasmacytoid. Where do you think this cell hangs around? In the plasma. Thank goodness, at least some microbiologists like us, right? So it's floating around in your blood. If it is floating around the blood, it is looking for something that might also be floating around in the blood. Some pathogens that might have infiltrated your blood, okay? So it looks basically for viruses that might be floating around in the blood, okay? So these plasmacytoid dendritic cells, they are special because they are capable of producing interferon type 1. Okay, so interferon is the first line of defense against viruses. Okay, that's a good thing to remember. First line of defense against viruses. And there are several types of interferons, mainly type 1, type 2. So this guy here is type 1 interferon, also known as alpha beta. Okay, so let's put a little note here saying that this is alpha beta interferon. Okay, type 2 interferon is interferon gamma. Okay, so this guy is type 1 interferon. Most cells of your body they are capable of producing interferon type 1. And we're going to see later why or how. Okay, but these guys here, they can produce a lot of it. Okay, and they are very good at killing viruses. Where is my pointer? There we go. They are very good at killing viruses and also at capturing microbes that invaded your blood. So those are the blood-borne microbes. Okay, now, once these cells catch the microbe floating around in the blood, these cells keep on floating. Okay, they are in your circulation, in your blood circulation, so they are not going to reach a secondary lymphoid organ. They are going to reach secondary. They're not going to reach a lymph node. Okay, because that's part of your lymphatic system, as we're going to see later today. Okay, so these guys here, they catch the microbe and they're floating in the blood, and then they're going to reach the spleen. Okay, so the spleen is a very important secondary lymphoid organ that filters pathogens that are floating in your blood. Okay, so the spleen is your primary blood filter. Okay, so then these guys here catch the pathogen, go to the spleen, and then it's going to present the pathogen to naive T cells, and it's going to also start ramping up the actions of your adaptive immune response. Okay, and in the process also, if pathogens are floating in your bloodstream and they get to the spleen, they can also be destroyed there. Okay, all right. And then you have this other type of inflammatory dendritic cells, and those here, they says they may arise from monocytes, and they are very much involved with the tissue inflammation in general. Okay, they produce cytokines that are pro-inflammatory, and there are few cytokines that you absolutely have to remember because they are very, very much pro-inflammatory, and they can cause a lot of trouble. So there are a few of them that when we get to them, you absolutely have to know them. Okay? All right. So here, you have cells that are derived here from your lymphoid progenitors, right? So they're called common lymphoid progenitor cells. And you can imagine if it is lymphoid, it's going to be B and T cells, right? So of course here you have your B lymphocytes, you have your T lymphocytes, and then there is a subset of these T lymphocytes that they develop into natural killer cells. Those are the NKT cells. And then you have other here innate lymphoid cells or ILCs, and those are going to develop into natural killer cells. It's a bit kind of confusing, natural killer T cells, natural killer cells. So let's just put them all in a bucket of natural killer, of killer cells, okay, natural killer cells. Okay. Now, how do you differentiate? I had mentioned that briefly before. How do you differentiate the lymphocytes, right? The different type of cells derived from lymphoid lineage. You have to look at the receptors that are on the surface of the cell. So this is the lymphocyte, looks round. This is the nucleus, or it's a big nucleus, little cytoplasm, and they all look the same. So here you can have a B cell, you can have a T cell, you can have a natural killer cell, and it's like, they all look the same. You look at the nucleus, it's like, I can't tell them apart, right? So the way that you're going to be able to tell them apart is based on some proteins that they have at the surface. And those proteins at the surface, they are called cluster of differentiation proteins, or CD molecules, okay? So in case here of a B cell, you're going to have some CD molecules like CD19, okay? So then in case of a T cell, you're going to have major classes of CD molecules. One of them is going to be CD4, and there are many other ones, okay? And then another one is going to be CD8, okay? Now, depending on the presence of either CD4 or CD8, then these subcategories of T cells are going to be different. If the T cell has a CD8, this one is going to be a cytotoxic T cell or a killer T cell, okay? If your T cell has CD4, it's going to be some kind of a helper T cell, okay? Now, keep in mind that before these cells, before they see any type of antigen, they are not active, okay? So they are all naive. Before seeing antigen, these guys are all, this is an N, is not an M, okay? Naive T cells. Never seen anything before. So they're quiet, not really doing any job, okay? Just waiting to see antigen. All right. So clusters of differentiation are important to distinguish the type of cells that you have. Okay. So here, the lymphocytes, right? So you have here, of course, your lymphocyte. You can have T cells, you can have B cell, And then look here as your cluster of differentiation molecule, right? So this guy here is right there. This guy is a CD4. This other guy here has a CD8, right? Now that cluster of differentiation is not the same thing as the T cell receptor, okay? Look, your T cell receptor is right here, okay, right there. So the cluster of differentiation is really close to the T cell receptor, but is not the T cell receptor. It's just something else that's on the surface of the cell, okay? And then here, when you look at your B cell, you're going to have the B cell receptor, but you're also going to have, what is not showing here, you're also going to have CD molecules that are on the surface here of your B cell. CD19 is a very typical one, okay? So besides your B cell receptor, you're also going to have CDs stuff on the surface of those cells, okay? Now, plasma cell derives from a B cell, okay? So a plasma cell is a B cell that has seen antigen, okay? So B cell, before it sees the antigen, what do you call it? You call it also naive, before seeing antigen. After it sees the antigen, what do you call it? After it sees antigen, it becomes something else. What do you call that? I heard it.
Killer?
No, not quite killer. B cells, we do have some killer bees, but we're not going to get to those. Huh? Better cells? Veteran! No, not quite. You call them effector. Okay? After they see antigen, they become effector cells. An effector cell is a plasma cell. That plasma cell produces antibodies. At first, the antibodies are on the surface, right? But then when it really becomes a plasma cell, then it's going to shoot antibodies out. Shooting antibodies out into the cytoplasm. And as we're going to see, there are many types of antibodies depending on how specific and how potent they are, okay? All right, so effector cell is not naive anymore, okay? And here you have an example of this natural killer cell that its job is to kill. And we're also going to see how that does its job, okay? So here there is this little diagram showing the connection of this cell lineage, right? How the common lymphoid cell goes into these differentiation processes. And keep in mind here that you have this T cell that branches into two types of T cells, and then cytotoxic T cell is just a cytotoxic T cell. But then the helper cell can become all different types of helper cells, depending on the pathogen that needs to be destroyed. Okay, and this is really, really cool, how each type of these T helper cells is going to be specialized in getting rid of a different type of pathogen. Okay, so here, lymphocytes, right? When naive, they all look the same. They have a big nucleus, very little cytoplasm, right? But then they see the pathogen, they see the antigen, and they're going to become effector cells, right? So looking here at T cells, they have those T cell receptors, that the T cell receptor gets to the T cell when it is developing in the thymus, okay? So during the development process, that's when the DNA of this cell, so DNA here, DNA of this cell undergoes recombinations, right? A lot of recombination, and is made to specifically recognize something that is not self, okay? So you have this process, as we're going to talk about, of selection of the cells that are useful, okay? All right, so types of T cells. So subdivision here of your T cells. So you have the T cells that have CD4 on the surface. So those are the CD4 positive cells, right? So your T cells are going to have either CD4 or CD8, okay? Either CD4 or CD8. I'm going to say all the time, but of course there are always some exceptions. So just keep in mind, in the circulation, you're going to have either one or the other, okay? Your CD4 positive T cells, those are your helper T cells, right? Your T cells. They are going to recognize antigen on a molecule that is presenting it to them that has MHC2 type of molecule. So that's called major histocompatibility complex type 2. Those cells are specialized on presenting foreign antigens to your helper T cells. Okay, so those MHC2 molecules present extracellular pathogens to your helper T cells. Okay, now, before seeing antigen, the thing is naive. After it sees the antigen, that thing is going to become an effector cell. Okay, what kind of effector cell it's going to become depends on what kind of extracellular antigen it's going to see. Okay, it has several options for becoming all different kinds of things. So it could become a Th1 or a Th2 or a Th17. Okay, this guy is actually a different degree of separation. So it could become one of those three types. Okay, so it is if it becomes. Why is it failing? Okay, if it becomes Th1 type of cells, they are going to have a main job at activating macrophages, okay, to better do their jobs. What is the main job of macrophages? To eat stuff and degrade them. Okay, and today we're going to see a little bit more on the mechanism of how they do that. Okay, they also have a job that is not very good on autoimmune chronic inflammation. Okay, some of these Th1 cells don't go away because of some problems with your immune system, and then you can develop some type of chronic inflammations. So that's also very much linked to Th1 cells. Okay. Now, Th2 cells, they are going to show up when your extracellular pathogen is some kind of parasite, okay? So these Th2 cells, they're going to activate eosinophils. Remember, eosinophils are the cells that are going to attack parasites, like worms and stuff like that, right? So Th2 is activated, is linked to function or better function of eosinophils. And they also activate mast cells. Okay. Now, Th17 cells, those are highly inflammatory T helper cells. Okay, so those are going to help activation of neutrophils. Remember, neutrophil is also highly inflammatory. So they're going to be like feeding each other on really boost up your inflammation process. Okay, so Th17. Now, this guy, of course, is involved in inflammation and is also involved in immune system disorders. Okay, so imagine a chronic inflammation. It's a lifelong process that only gets worse. Once you trigger chronic inflammation, it's like it doesn't really go back. It just keeps on getting worse and worse. Acute inflammation is good. You need that as a main immune system type of defense. But chronic inflammation is just a pain. Okay, I mean, literally and figuratively. Okay, so these other guys here, these Tregs, they are also CD4 positive, but those are the peacekeepers, right? Those are the ones that are going to quiet everybody down. They're going to suppress the immune response, and they're going to help with the development of tolerance to your own cells, tolerance to things that are not harmful to you, right? So Treg cells is just a whole different category in itself, but they are CD4 positive cells. Those are the ones that are suppressor cells. Okay. So here are the guys that are CD8 positive, those are your cytotoxic T cells. They are going to recognize antigens that are presented on major histocompatibility complex type 1. Very important, type 1, okay? That means type 1 is going to present intracellular pathogens. Give me an example of an intracellular pathogen. There's a very common viruses, right? Or some other strange things that can happen inside the cell, okay? So MHC1, it's going to present intracellular pathogen. What CD8 is going to do is going to kill the presenting cell. No more questions asked, okay? It's going to recognize something that is foreign, it must be some pathogen, so I'm just going to kill the cell before that pathogen starts to replicate. Okay. Yeah, very much. And then we get to this kind of problem that sometimes the thing that he sees is really a bad pathogen, right? But then some other time it's not. But then it already killed the cell, so it's not like it can go back, right? So there might be problems with that kind of recognition. But that's what it does, okay? Another problem is that these cells also attack transplanted organs and tissues. Okay, this is the cause of tissue rejection. Okay, so when you have organ transplant, first thing that you have to check is for compatibility. And when you check for compatibility, you're going to look at those MHC1 molecules. They should be compatible between the donor and the recipient. Yes. MHC1 is in all nucleated cells of your body. All cells that have a nucleus, they have MHC1 on their surface, okay? Including all the organs, okay? Now, does a red blood cell have MHC1? No, it doesn't have a nucleus. So that's a nucleated cell, no MHC1 there. This is why this process of the immune system does not attack your red blood cells. That's why you can do blood transfusions just by matching ABO types, okay? And Rh factor positive or negative. Okay. All right, so those are your cytotoxic T cells. Then you have your B cells that, of course, they are subdivided into several categories, okay? The B cells, once they mature, they become effector cells. That's when they produce antibodies, right? They also undergo clonal selection during the development, okay? And they undergo a further modification after they are activated, okay? Because they have to produce the antibodies, and they have to shoot the antibodies out of the membranes and into the surrounding liquid, okay? So they also undergo some type of change after antigen presentation. That's when they are plasma cells, okay? And then some of them, after they see plasma cells and after they have the clonal expansion and then the clonal retraction, some of them become memory cells. And then they go to the bone marrow and they sit there until they are needed again. So some of them stay there like forever, for your whole lifetime. You have memory cells against some antigens. Okay. So you have mainly two subcategories of B cells. You have B1 subtype, and that is present mainly in mucosal tissue. Okay, so they're very important in producing antibodies in the mucosal tissue. The types of antibodies that they produce in mucosal tissue is mainly, immunoglobulin A. It could also be some IgM, okay, but it is mainly like IgA, okay? So those are very important for mucosal function, as we're going to see later, okay? All right, so the B1 subtype also, look at this, this is not a coincidence. They are going to produce the antibodies. They're going to recognize antigens on the surface of your red blood cells, the AB group antibodies. Okay, keep that in mind. because for a long time I had no idea how we develop antibodies against broad group antigens if the baby was never primed against the different blood groups. We'll get more to that, we'll never get to that, right? But look, this B1 subtype is the one that produces the AB group antibodies. Okay. Then you have this other subtype that is the B2 subtype. That is the type that is in your bloodstream and in the lymph. Okay, so that's the type that is floating around, not really quite associated to mucosal tissue. That is B1 with mucosal tissue. So B2 is everyone else that is in the lymph nodes and in your spleen and in the circulation. These are all B2 cells. Remember, they are naive, and then they start producing antibodies. When they are naive, they have some antibody that is stuck to their surface, as we're going to see later, these types of antibodies, right? And then when they become effector cells, they are going to class switch their antibodies, and they're going to start producing antibodies that are specific to the type of infection that they encounter. Okay. Cell. Now, these B2 cells, they are also capable of phagocytosis, okay? So they are capable of picking up some bacteria that is out there, okay? And they are capable to present it to T cells. But they are not very effective at presenting this to naive T cells. They might present it, but then the naive T cell is not really quite, it's not going to be quite activated with a B cell presentation. But if this T cell is not naive, okay, if this T cell is, for example, a memory T cell, then your B2 subtype can activate the T cell that has seen previously an antigen and has previously been activated. Okay? All right. So then here you have your natural killer cell. Okay? This guy does not have an antigen receptor. Okay, does not have an antigen receptor. He has an IL-2 receptor, and as we're going to see later, interleukin-2 is important for activation of cell division, okay, so proliferation. So this guy has IL-2 receptor, and what they do is that they kill infected cells and they kill tumor cells, okay? How is it that they detect infected or tumor cells? Well, the infected cell, let's say this guy is an infected cell, okay, so here is a virus here. When the virus is inside of the cell and it starts to replicate, some proteins of this virus, they're going to downregulate this molecule on the surface of the cell that is MHC1. Remember, all nucleated cells have MHC1, right? So when the virus is there, what's going to happen to that MHC1 is going to disappear from the surface, okay? So now your natural killer cell goes there. So this is your natural killer cell. It is looking for cells that don't have MHC1, okay? So it gets there and it does not detect MHC1. So now it's going to kill this cell, okay? So imagine now that happens when you say the cell is looking for something else. These cells don't have eyes. They have to go with receptors blindly, okay? They're like this. They go and go and go. Oh, you have MHC1? Good boy. You have MHC1? Oh, good. You survived. You, no MHC1? I don't care who you are dead. That's how they act. Okay. So, simple. No MHC1, you get killed. Okay. Okay. All right, so here we go for more functions on how these cells are doing their job. Okay. So as you mentioned, one type is your natural killer cells, and once they detect the cells they have to kill, they're going to kill. How they're going to kill? There are two ways that these cells kill, as we're going to see again, in more details later. They can use a fast ligand pathway. Okay, so fast activator and fast receptor. And what happens is that at the site of contact, this Fas ligand that is present inside your target cell, it gets connected to the Fas on the killer cell, and it triggers a whole process of killing this cell from inside. Okay, so it triggers apoptosis of the recipient cell, or some type of cell death. Okay, more accurately, pyroptosis, but it's death from the inside. Okay. Another way that these cells can kill is by enzymes. So we can use granzyme or perforin enzymes to literally punch holes at this target cell and make it leak out its contents, punching holes. Okay, so that's another form that natural killer cells can kill. Okay. All right, so here you're describing functions of the adapted cells. Okay, you have your helper T cells. Those are guys that are going to activate macrophages, and they are going to activate also cytotoxic T cells and B cells. Then here you have your regulatory T cells. Those are your Tregs. They are going to suppress everybody. Okay, you had a question? Okay. And then you have here your CD8 positive cytotoxic T cells. Those are going to kill infected cells. Okay. Now remember, lymphoid cells, you have your B cells. They progress into plasma cells. The main job of plasma cells is they produce antibodies. Okay. So how is it that the B cells develop in the primary lymphoid organs? Basically, the B cell is going to develop in the bone marrow. That's why B is for bone marrow. Okay? So those are going to develop not because they just decide to develop into B cells, but because the progenitor cell, the pluripotent stem cell, is in contact with other cells in the bone marrow, and those cells are called stromal cells. Okay, those are your stromal cells. And these cells produce signals that are going to tell the progenitor cell to start expressing some molecules, some proteins that are important for B cell development. Okay, so the stromal cell is going to allow this hematopoietic stem cell to proliferate, and is going to direct protein synthesis and subsequent migration of that progenitor B cell into areas of the bone marrow that are going to further differentiate that initial cell into a functioning B cell. Okay. So here you see a picture. This is your bone. Okay, so those are cells. Those are bone cells, and here is your bone marrow. Okay. So here you have a big blood vessel, and then you have endothelial cells that are close to your blood vessels, and this guy here. Okay. So endothelial cells here that are surrounding the blood vessels, and then you have here all this mash that is in your bone marrow. Remember, this is your red bone marrow, right? And then you have your progenitor cells. Now, all this mash here, you have these other cells that are not showing here, these stromal cells, that are producing these factors, okay, cytokines and growth factors. And those are coming into contact with your pluripotent stem cells, and it's going to allow them to differentiate. Okay. So here, how is it that T cell develops? So B cell develops all in the bone marrow. Now T cell is a bit more complicated. They start to develop in the bone marrow, okay? And then they're going to leave the bone marrow, and they're going to migrate to the thymus, which is right there. Okay, here is your thymus right here. Okay? So don't confound this with your thyroid. Thyroid is here, thymus is under there. Okay, it's like on top of the heart. Okay, so this is your heart right there. Okay, so here's your thymus. Okay, so these cells migrate in the blood, they go to the thymus, and then in the thymus they are going to further differentiate. They're really going to be tested whether they can detect the self-proteins or not. Okay, so if they detect self-proteins, they're going to be killed. If they do not detect self-proteins, then they're going to be allowed to leave the thymus. Okay, so that is going to happen in the cortex and medulla of the thymus. Okay, so in those places inside of the thymus, you are going to check the affinity of the T cell receptor. You're going to check the affinity for MHC molecules, because remember, T cells are going to recognize stuff that is presented on MHC molecules. So first thing that they have to do is check if they can recognize MHC molecule in the first place. Okay, so if they recognize it well, then they are selected, like for positive selection. Okay, if they don't recognize MHC, they go through negative selection. They are killed because they're useless. Okay, recognize just the right way, they're positive selected. Don't recognize it, they get killed. And there is more to that, but this is a simplified version of how they get selected at first. Okay. So here's a brief look at the human lymphatic system. As we know, we have two forms of circulation in the blood. Oh, that reminds me of now in the chalkboard. We don't have chalkboards anymore. Like, yeah, anybody seen a chalkboard and like, okay, that worked, yeah. Okay. So here you have circulation, right? You have here your blood circulation, right? It goes to the arteries, it goes through capillaries, it brings oxygen to the tissues, it picks up carbon dioxide, it becomes blue, and then it goes to the veins, and then it goes back to the heart. So that's the blood circulation. And that's not the whole story, right? Because when the blood passes here to these very, very fine capillaries, some of the liquid that is part of the plasma, some of the liquid leaks out of the blood. It goes into the tissues, okay? And that's not good, because if it sits there, then whatever tissues have this extra liquid, it's going to start swelling, okay? And you might have noticed, especially with the older population, like if they're standing on their feet all day and then you look at their feet at night, it's like looking like a balloon, right? It's very swollen, right? Because the whole circulation is not as effective as with you guys, young people, right? And you can also notice during pregnancy, because the rest of the body has a lot of pressure and a lot of these veins are being compressed by this baby growing in that belly, right? So by the end of the day, a pregnant woman can't stand even walking. You gotta put your feet up, right? Big problem. Okay. So this liquid leaks out of your capillaries, but they're not going to be sitting there forever. This liquid has to be reabsorbed, okay? So this is when your lymphatic system comes into play. So you have here lymphatic capillaries that they have open ends at their ends right there. Okay, so let's put here little holes. They're open ends, okay? And they're going to basically suck up that liquid like a straw by capillary action, okay? So the liquid naturally comes out of here and is drawn like a straw by capillary action to these lymphatic vessels. And then this liquid is drained here, drained there, gets together to bigger and bigger lymphatic vessels, and they circulate back and they are dumped here into the vein. Okay, it's like the vena cava or something up there, okay? In the process, this liquid passes through lymph nodes. There are these little dots here that are in the middle of those lymphatic vessels, okay? So when this liquid passes through the lymph nodes, this liquid gets filtered for the presence of possible pathogens, okay? So then, you have, so let's see here in the, when you look here, this area close to the clavicles, right? To the clavicles, you have these veins here. One of them is at the left side of the body. So this one is facing us. So here is our left side, right? You have this left subclavian vein. Okay, so that's important here. Your left subclavian vein that gets all this liquid that came from the left side of the body. And then you have here a right subclavian vein that collects all the liquid here from the lymphatic ducts that came here from the right side, so from the left side. And those are also going to collect from the head too, from the size of the head. Okay. So let's see here. Okay, they're different. Okay. Now, just for recap here, you have a good slide for your organs that you have here primary organs, very easy to remember, right? And then you have your secondary organs, and then here you have your lymph nodes and your spleen, and something we didn't talk much about, your mucosa-associated lymphoid tissue, okay? And that's abbreviated MALT, mucosa-associated, and they have all kinds of subcategories of MALTs and BOUTs and whatever else, okay? These are important for your defense at the mucosal areas, okay? Other things that make your lymphatic system, of course, lymph vessels, right? And one thing that is really, really important to know is that your secondary lymphoid tissue is the site where your innate cells activate your adaptive cells. Okay, so that's the site where all the action is taking place. Okay? Keep in mind also secondary lymphoid is the spleen and also your lymph nodes. Okay, so here they are. So you have your primary and then your secondary, right? Lymph node and spleen, and then your mucosa-associated lymphoid tissue. Those are your secondary. Okay, so let me see what is important here. System. So we talk about lymphatic system. Oh yeah, places that you find a lot of lymph node is like on your neck, under your armpit, and inguinal region, like kind of like groin area, you know. And this is interesting because when you go sometimes just for a general office visit, sometimes the doctor is going to palpate under your lymphoid, like by your armpit, to see if you have any enlarged lymph nodes, right? That could indicate some kind of a problem with cells that are nearby, like for example breast cancer cells, right? Right. So yeah. So we're going to see also the relationship of why you have these lymph nodes distributed all over the body and what they mean, what it means when some lymph nodes get activated and swollen up, right? So it means a lot of adaptive immune system trying to kill something that is not supposed to be there, right? All right, so the spleen is going to filter the blood. Okay, filter for blood-borne pathogens. It's also important for recycling of blood, red blood cells. Okay, it's going to kill defective red blood cells, and is going to serve as a reserve site for platelets and for more red blood cells. Okay, so your spleen is right there. Then you have your thymus gland, and this is when you're going to teach your T cells how to behave. So that's your primary. This is secondary, and this is primary. A little bit less innate immune type of catching those cells. Yeah. But then it's not really critical because you have all these other backup mechanisms that are going to really take care of any kind of pathogen that goes to the mouth. Okay, I can tell you a little story about that too. Okay. So here you also see your Peyer's patches. They are secondary lymphoid tissue that is very, very important for your intestines. Okay.